Hello, everyone. Thank you, Catherine, for the invitation and the introduction. So as Catherine mentioned, my name is Jaime Dimant and the Director of Application at InVicro. And I'm going to be presenting today a few case studies, examples of how we've been utilizing ZIRA, CFT, in a way to maximize data collection from some of our preclinical work we've been doing in InVicro. We do a lot. I'm going to be discussing about some multimodality studies, but we've really been using ZIRA and CFT approach in a very useful way in many of our multimodality studies, and I'm happy to present the work we've been doing. So my talk will be basically divided to three, briefly going over the preclinical imaging modalities, the common one, how they complement each other, where they complement each other, be discussing about the volume to resolution trade-off. This is where, again, the complementation really comes in handy because you'll see there's going to be a trade-off between sample volume and resolution, which will then lead me to Cryo-Fluorescence Tomography, the technology itself. I'm going to be briefly introducing the technology and the data output, what eventually we typically get from CFT imaging. And then we'll be really focusing on case studies. I'm going to be going through three specific ones. The first one looking at AV distribution versus transduction, a question very important when we're doing gene therapy development. Looking at ASO pharmacology, so a very broad study where CFT was part of, looking at all the way from PK, PD, all the way to microscopy on, again, all of those case studies, again, same animals. So really where we maximize the data collection. And lastly, cell tracking. So you can see here, listing out six very common imaging modalities for rodents in preclinical work. I'm not going to go through these. You see they're listing advantages and disadvantages. And really the point of this slide is to emphasize that each imaging modality has its advantages and disadvantages. In many cases, they're not competitive. They're really complementary. So you can achieve you can answer one scientific question with one imaging modality, but not another. And then you can potentially include another imaging modality to answer that question, which you cannot with the other. For example, if you look at optical imaging, the disadvantages of optical imaging, the limited clinical translation and the low depth penetration is the advantage of SPECT imaging. So I will be showing a study where we've done SPECT imaging to facilitate PK, to provide some information, but then also added another layer of data by conducting Cryo-Fluorescence Tomography. Now, if you take all those imaging modalities and you plot them on a resolution to mass of tissue graph, as you can see here, you'll see that the in vivo imaging modalities, bioluminescence, fluorescence, nuclear modalities, allow you to image large sample volumes, so whole animals. Resolution, however, is not high. You're in the kind of millimeter resolution. On the other hand, if you're looking at cellular microscopy, then you are able to increase your resolution to cellular resolution, but then you're limited in the mass and the volume of tissue you can image. There's basically so many sections one could image. You're limited to sections, and you'll have to, in a sense, kind of pick from which regions you're collecting sections, and then you can conduct cellular microscopy. In between, there's a void, basically, where CFT really fits in. It allows you to image still large samples. We can image whole samples, whole animals, many organs, obviously whole organs, but more than one at one go. But provide you with much higher cellular resolution. So much higher resolution, still not cellular resolution, but really allow you to get closer to the cellular microscopy. So really kind of bridging between the in vivo and the microscopy. And in many cases, you can combine. So if you're doing, for example, in vivo fluorescence, then you can, of course, do Cryo-Fluorescence Tomography and then all the way down to microscopy. And these are the case studies that I'm going to present how we, at InVicro, how we combined these approaches together. So specifically discussing the kind of multimodality approach for optical in vivo imaging. Generally, optical in vivo imaging is a very powerful tool for preclinical research. Here, I'm specifically kind of touching on gene therapy and cell therapy. It provides early understanding of kinetics and distribution. It's pretty economical. It's not that expensive, and it's very widely used. It's widely used because it's obviously simple and easy to operate. A lot of labs, a lot of companies are using IBIS, for example, in vivo fluorescence or bioluminescence, and it's very versatile. The advantages of optical in vivo imaging is that it's in vivo, it's cost effective, and you can image multi-sample, basically you can image several animals at once. The disadvantage is the resolution, which is not high. It's most cases just 2D. Basically, you're imaging the face of the animal. And then for fluorescence, more than bioluminescence, you have high background and low signal to noise. So if you look at common biomarkers for gene therapy, so luciferase is a very common biomarker for gene therapy, where you would introduce luciferase and be able to kind of screen and see where you're seeing transduction of your protein. For vector cell tracking, NIR conjugates are also widely used, where you would, again, conjugate ideally an NIR fluorophore to track the vector of the cells. And fluorescent proteins also very commonly used as a biomarker for gene therapy, where instead of luciferase, you will have a fluorophore, typically GFP. If we speak about GFP is probably the least favorite fluorophore for doing fluorescence in general, but certainly in vivo fluorescence. You can see in this slide here, with the really fluorescent protein in general, but definitely GFP, they're really suboptimal for optical in vivo imaging. There's really low tissue penetration definitely in GFP. The more you go to NIR, you're gonna be able to penetrate more into the tissue and potentially detect, but still, it's a really big limitation. Obviously, there's attenuation of emitted signals. So even if you are able to penetrate into the let's say deep fluorophores, there's a very good chance that the signal emitting out will get attenuated. And as I mentioned, there's autofluorescence. So if you look at fluorescence compared to bioluminescence, the autofluorescence is a big problem. As you can see it here, there's a lot of background if you're doing in vivo fluorescence, not as big of a problem, or not a problem when you're doing bioluminescence. And this is how in vivo fluorescence basically look like. This is an example it's a tumor-bearing animal. I'm going to be going in more detail about the actual case study itself and what we did here, but I just wanted to kind of pique your interest and show you how an anesthetized live animal is looking as we're imaging basically we're looking at cells here at macrophages in a tumor. Now, if you take the same animal, once you conduct the in vivo imaging and you sack the animal, conduct Cryo-Fluorescence Tomography, this is how the same animal looks like. So again, we're looking at macrophages here. I'm going to be going into more detail that later on, but that's the exact same animal. You're seeing the tumor down here, as you can see it here. But again, it's closest to the surface, which is why you're able to see it with in vivo fluorescence. The liver, which is very hot, we were unable to detect it in this case. We're also seeing uptake in many more organs, bone marrow, lymph nodes. So again, I think this is a nice example for how we can add or reveal more information as we're doing Cryo-Fluorescence Tomography on top in vivo fluorescence. And for the potential skepticals, also autofluorescence, something we are aware of and always control for. So this is a mouse, just a control animal, no autofluorescence. In this case, it's size 7. So there's no size 7 in this animal. The only signal we're seeing is autofluorescence coming from the gut, which is due to the chow, the chlorophyll in the chow, which we can potentially minimize by feeding the animals alfalfa-free chow. So the way we image this animal is using Cryo-Fluorescence Tomography. It's a Blockface Imaging modality. You can see on the left a video showing a whole mouse frozen. There's no fixation. There's no special processing. You just freeze the animal, block it in OCT. We install fiducials, those red dots that you see, those black dots that you see here. Now we have a blade that will basically, as you can see here, will come and section off a section of the block. Typically, 25 to 50 micron is what we work with. We have an optical setup placed above. It's out of, you're not seeing it here, but it's straight above the block. And we basically conduct it in a sequential manner. So we image the remaining block, we section, image the block section, so hence block face imaging. We do that throughout the entire sample. Each plane, we collect the fluorescent signal as well as white light. As you can see, since we don't perfuse, we don't fix, we really are able to maintain morphology. So we collect the data. We're imaging both in the white light and fluorescent data. Eventually, we reconstruct the data. I'm going to show you how the output looks like. On the right, you see the Zira itself, the instrument. It's basically a very big cryomacrotome. The animals at the block are placed right below this tower that you see there. This is where the optical system sits, and it's basically completely automated. You place in the block with the sample, and you've determined which channels you want to use, what tissue thickness, for example, and then you walk away. Eventually, processing and depleting the entire sample, the entire block. The typical output looks like that. You see on the bottom left here a whole MIP of a AV9 administered AV9 expressing GFP in a mouse. So again, you see GFP. So the ability to detect such resolution and such penetration is very impressive. You'll see the whole liver, for example, lining up with GFP. You see the heart, and I can show you the heart is here, but if you play this movie here, so that's the second output, the fly-throughs, where we overlay the fluorescent with the white light. So here you see the liver, you see the heart starting to show up here, for example. Another really cool thing to see here, to note, is AV9 have they have high tropism to dorsal root ganglions. You can see here along the spine, these dots here. So the ability to detect and resolve DRGs, again, very useful. Lastly, we keep our voxels isotropic, so we're able to reconstruct the data and view it in any rotation we want. So here is an example for a multi-planar visualization software where we are basically able, once we reconstruct the data, we don't have to necessarily be limited to the section view. The section view, we can then image in any view or orientation we want. Here is another quick slide that I want to go through just to briefly speak about the volume to resolution trade-off. We're taking one image of the entire field of view. So bigger field of view, resolution goes down. We have a lot of flexibility in the type of samples we image, from going all the way from, on the right, a mouse kidney, a rat brain, or a whole head. So again, we have fairly large blocks, so we can image a whole cyno head and basically reconstruct the data and create these nice fly-throughs. So finally, I want to go into some case studies. The first one is looking at the AV biodistribution versus AV transduction. So here is a study we've done together with Voyager Therapeutic. We have shared and presented this work. Here we're looking at the pharmacokinetics of the AVs. So we've wanted to look at the kinetics of AVs following intrathecal administration. We conducted iodine 125 labeling, and then they inspect images. So the rats were administered with the labeled AVs intrathecally. You see in a very fast, very early time point, you're seeing rostral distribution from the lumbar region where the AVs were administered all the way into the head. you're looking at about the 29 hours. By the way, as soon as you start seeing signal in the thyroid, you know that the iodine is no longer attached to AAVs. But up till that time point, we are able to basically inform about the distribution of the AAVs. Once we've completed the spec data, we did not tag the animals. We did not complete the study. We basically allowed the animal to recover. The AAVs that we administered were a mixture of labeled AAVs and AAVs that expressed a fluorescent protein. And for that, we allowed the animal to recover for several weeks, waited for the fluorescent protein to express, and then we collected only the vertebra. Again, the idea is to look at the distribution, looking at the spec data of the vector, looking at the spec data versus the actual transduction. So this is how the vertebra looked like after Cryo-Fluorescence Tomography. You see here the reconstructed MIP of the signal surrounding or in the vertebra. And again, when I first saw this data, to me, it looked like the signal is not in the, although we were able to confirm based on spec data that the AVs went all the way into the head. So the CSF, the IT administration was successful. We're seeing signal that seemed to me outside. This is where the overlay became very handy, because if you look at the overlay here, it's becoming more clear that the signal that we're seeing outside the vertebra, it's outside the vertebra in surrounding tissue. And we were further able to, so this is another thing we basically did, again, to compare head to head. We've co-registered the CFT data with the spec data into the same volume. So again, these three modalities here co-registered in the same volume, as you can see in the right here. So spec is in green, CFT is in yellow. And you can see the discrepancy between the distribution of the AVs and the eventual transduction. It was a very again, useful and interesting results. And again, to kind of confirm that the signal that we're seeing is outside the vertebra, we collected sections. So with Cryo-Fluorescence Tomography, we are literally, it's not computed tomography, it's tomography per se, meaning we are collecting each and every plane. We collected sections, and under a microscope, we're able to confirm. You can see here the vertebra in blue here in the middle of the spine. And if you look outside, there's confirmation that the IRFP expression is outside the vertebra in surrounding muscle. The reason for that, we in hindsight figured out, is during that time where the animal was allowed to recover and express the protein, there was basically a catheter stuck in his spine. So we didn't pull it out. So what likely happened, some AVs just kind of leaked out around the lumbar area where we administered the AVs and just transduced surrounding tissue. So again, very useful from the same animal to be able to, so in summary, basically, we've conducted a PK study looking at the distribution, the kinetics of AV distribution using SPECT-CT. We then combine that with Cryo-Fluorescence Tomography to look at the spatial transduction of the AVs. Again, looking at a much higher resolution compared to SPECT-CT. And then lastly, we're able to use microscopy to look at the in a sense, the transduction in a high resolution. So another case study that I would like to present is a work that was published with work we've been doing with we did with Biogen and Ionis, looking at ASO pharmacology. So here again, similar in a sense approach, we've used nuclear imaging to look at kinetics. In this case, also utilizing SPECT-CT and iodine-125, ASOs were radio-labeled with iodine-25, intrathecal administration into rats. Again, very kind of like with the AVs, supporting the data we've seen there, very fast rostral distribution over a short period of time. And then if about 4 hours, you're seeing rostral distribution all the way to the head. Then at 4 hours, we're starting to see clearance into lymph node, cervical lymph node, and kidneys, all the way to day 7. Basically, on the left here, you see we just kind of combine all those scans into a single volume, so you can see what happens over time. Again, the distribution and the clearance. Here on the right side, that's not SPECT, that's AutoRAD. So we've also combined AutoRAD in this case to increase the resolution, looking at one single plane. Again, with AutoRAD, here you're looking at a single plane of the to look at the distribution of the ASO. The question here, though, was: are we able to confirm that the ASOs, once reaching the head, they're penetrating into the parenchyma? Are they getting into the brain, and which cell types are they ending at? You cannot say that or answer that using SPECT-CT. The resolution is not enough. And most likely, a lot of the signal that you're seeing here up in the head is in the meninges. You cannot really say or resolve whether the signal is in the parenchyma or not. This is where we've, again, combined Cryo-Fluorescence Tomography. Instead of iodine 125, we've labeled the ASO with size 7. We did a similar study, and it's, a PK. I'm doing this because it's each of these time points here is obviously a separate animal. We have to take down the animal, but the time points that we've determined here were based on the SPECT study. So the SPECT, the PK study informed us which time points we want to take animals down. We administered the animals same dose, intrathecal ASOs labeled with size 7. In this case, only collecting the brains, again, to minimize the field of view, maximize sensitivity and resolution. And here you can see, again, in a much higher resolution than with SPECT, the distribution of ASOs in the brain. You can see that in the earlier time points, a lot of the signal is kind of around the meninges. You can clearly see here blood vessels that have positive signal. And then over time, you can see that the signal one goes down and becomes more diffused. So it's going down, which is in agreement with the clearance that we've seen over time. The diffused signal is suggestive of the fact that the ASOs are no longer only in the blood vessel in the meninges, they're starting to penetrate into the parenchyma. On the left here, you can see again just an example for the kind of multi-view functionality, a single plane, several views over time points to appreciate the fact that in 1 hour, most of the signal is more kind of around the meninges. As you go into the 8 and 12 hours time point, the signal covers more tissue than it is in the earlier time points. And further than the CFT data, which allowed us to, again, suggest or indicate that we are seeing penetrance into the brain, we collected sections and conducted immunofluorescence. Here, the sections were collected on glass slides from mid. So we start the CFT process. We've collected sections and run several immunofluorescence assays to basically characterize or identify which cell type, in which type of cells the ASOs are ending up: neurons, macrophages, microglia. Here is an example for a few actually, SMA and RECA1, again to understand along which vessels or how the ASOs are potentially getting into the parenchyma. Here you can see on the right, and I apologize, the ASO here is in green and here is in red, so it's all digital, but you can see on the right side, ASOs in green, a lot are still in the meninges, again, confirming what we've seen with CFC, confirming what we're suggesting is happening with the SPECT data. A lot of the signal is in the meninges, a lot of the ASOs are in the meninges. But if you look at the stain here, the NUN stain, which stands for neurons, neural nuclei, you can see that we have colocalization. We're seeing green ASOs, so the size 7 labeled ASOs co-localizing with the neuronal nuclei, confirming that the ASOs are getting into the parenchyma and are eventually ending up in neurons. Another thing we did here was also a PD, a pharmacodynamic study using using PET, using a PET tracer for GABA1. The ASO here was basically it's a GABA1. We're doing GABA1 knockout. Flumazenil is a GABA tracer. So if you look at the bottom panel, you're seeing that following treatment, there is reduction in flumazenil uptake, meaning the GABA receptors are being knocked down by the ASOs, as opposed to the control, the MALAT1, which is just a housekeeping gene, in this case, a negative control, showing that when you administer the MALAT1 ASOs, flumazenil, obviously, there's no reduction. And this is, you're looking at the GABA uptake in a non-knockdown animal, basically. So to summarize, again, we've combined here in one or two cohorts. We did PK, looking at the distribution of SPEC data, looking at the distribution of the ASOs, then combined it with PET data to look at the PD, to look at flumazenil uptake, then added CFT to understand in high resolution the spatial distribution of the ASOs in the brain, and confirm with IHC down to the cellular level where the ASOs are ending up at. So the last case study that I would like to present is macrophage tracking. Here, the objective was to determine whether baseline macrophages are detectable in xenograft models. So the idea was to prepare to a follow-up study where we wanted to see potential macrophage recruitment into tumor, but we wanted to see what is the baseline macrophage level in a whole mouse. We've combined here a test article that is a vSense, commercially available. You administer that in vivo. Macrophages would uptake those molecules and will basically be allow us to track macrophages in vivo. These vSense are dual label. They have both fluorine 19 for MR and size 7 for any optical imaging you may want to do. And here, just on the bottom there, you see that we first administered the turpentine or PBS. So we've created a positive control by administering turpentine, created this macrophage recruitment, and I'll show you how the data look like in the turpentine, for example, versus the PBS, which would be the baseline animal. So looking at the MR data, you're looking at a lot of uptake in the liver, which totally makes sense. There's a ton of macrophages and Kupffer cells in the liver, so they're uptaking the vSense, those nanoparticles, and are residing in the liver. You are seeing in the turpentine sample, the positive control sample, tumor. So these are obviously tumor-bearing animals. So you're looking at signal in the tumor in the turpentine, the positive control animal. Looking at the PBS animal, you're also, looking at the PBS animal, you're looking at the, again, a lot of liver uptake, but we are able to detect here vSense in the tumor as well. So baseline tumor minus turpentine, just tumor-bearing animal, we're able to detect macrophages in the tumor. And on the right, it's just a negative control for basically just a PBS-administered animal. Now, if you take the exact same animal, this is, again, the same animal that I presented several slides ago. If you take those same animals after you're doing the MR, we've also kept them anesthetized, conducted IBIS imaging. And here, again, you're looking at the turpentine signal versus the control signal. Increased, you're seeing more signal in the turpentine, but both animals, you're seeing the signal in the tumors. And here is where I want to just emphasize that you're not able to detect the liver, right? The liver that you saw in MR, we know there's a ton of signal, there's a ton of macrophages and vSense is in the liver as well. We're unable to detect it in this case using in vivo fluorescence because of all those limitations that I've described, penetration, depth of penetration, and signal attenuation. Now, if you take those same animals and conduct Cryo-Fluorescence Tomography, this is the type of data you see. Here is and I think it's also a nice example to show the multi-channel, basically the spectral options for Cryo-Fluorescence Tomography. There's several channels. Here we're looking at the GFP signal of the tumor cells. So the MC38 cells in this case express GFP. So again, a lot of, as I mentioned, a lot of autofluorescence coming from the gut, as you can see basically in all the animals. But if you look at the tumors on the right, on the side of the flank of the animal, you can see we are able to detect the tumor. We're seeing GFP fluorescence in the tumor. Now, if we image for, I think I'm missing a slide here. Well, I'm missing one slide here. But eventually, we're able to take all the tumors. So this is all the tumors that were imaged in the same animals. In this case, we just crop them. Again, we're imaging the entire mouse, but we're able to crop only the tumors and look at the macrophage signal versus the MC38 signal, basically the tumor. And eventually we're able to co-register the VSense and the GFP signal to really understand the spatial distribution between macrophages and in a sense viable. This is again looking at the crosses versus not. Obviously the viable cells will express GFP. So looking at macrophage distribution versus tumor viability. I believe I can stop here and just again summarize this specific. Again, this is some reason I'm missing. I was missing the slide of the actual VSense images. But again, this is the baseline mouse here. You see again, besides the tumor, as I mentioned, you're looking at lymph node uptake, liver uptake, which all makes sense given that we're looking at macrophages here. And to summarize also, again, another example for how we've maximized data collection by conducting three imaging modalities on a single animal cohort. So we started with the MR to look at the kinetics of macrophages. In this case, we haven't done a lot of time points. It was really kind of a proof of concept that we're able to detect. We then, under anesthesia, conducted again kind of tumor PK, looking at the PK signal, at the Cy7 signal in the tumors, still using in vivo fluorescence, and then lastly, looking in a higher resolution and higher sensitivity. Again, with Cryo-Fluorescence Tomography, it's not only about the resolution, it's definitely about the sensitivity, the fact that we are sectioning the animal, we're exposing each and every plane. There's no signal attenuation, there's no body attenuation because there's no body. We're literally exposing each and every plane, maximizing sensitivity, and as you can see here in this example. I will stop here and thank you everyone for your attention and happy to take any questions.